Dear students, Assalamu alaikum. From today, we will start the study of acid-base regulation. In the next few lectures, we will consider the different mechanisms regulating hydrogen concentration and also reabsorption, production, and excretion of bicarbonate ions, which is one of the key components of acid-base control systems in the body fluids. The objectives of today's lecture are to learn about definitions used in acid-base regulation, systems regulating hydrogen ion concentration in the body fluids. Hydrogen ion concentration is precisely regulated because the activities of almost all enzyme systems are influenced by hydrogen ion concentration. Before beginning today's main topic, let's revise some of the important definitions which will be used quite frequently in the discussion. A hydrogen ion is a single free proton released from a hydrogen atom. Molecules containing hydrogen atoms that can release hydrogen ions in solutions are known as acids. A base is an ion or a molecule that can accept a hydrogen ion. The terms base and alkali are often used synonymously. An alkali is a molecule formed by the combination of one or more of the alkaline metals with a highly basic ion, such as a hydroxyl ion. Alkalosis refers to excess removal of hydrogen ions from the body fluids. Acidosis refers to the excess addition of hydrogen ions to the body fluids. Strong and weak acids. A strong acid rapidly dissociates and releases large amounts of hydrogen ions in solution. A weak acid slowly dissociates and releases less hydrogen ions in solution. Strong and weak bases. A strong base reacts strongly with hydrogen ions and quickly removes hydrogen ions from a solution. A weak base reacts weakly with hydrogen ions and slowly removes hydrogen ions from a solution. Most acids and bases in the extracellular fluid that are involved in acid-base regulation are weak acids and bases. As hydrogen ion concentration is very low, it is customary to express hydrogen ion concentration on a logarithm scale using pH units. A low pH corresponds to a high hydrogen concentration. A high pH corresponds to a low hydrogen ion concentration. The normal pH of arterial blood is 7.4. A person is having acidosis when the pH falls below 7.4 and is having alkalosis when the pH rises above 7.4. Intracellular pH usually is slightly lower than plasma pH. The pH of intracellular fluid has been estimated to range between 6.0 and 7.4. The pH of urine can range from 4.5 to 8.0 depending on the acid-base status of the extracellular fluid. Defense against changes in hydrogen ion concentration. Three systems regulate the hydrogen ion concentration in the body fluids to prevent acidosis or alkalosis. These systems are: number one, buffer systems of the body fluids; number two, respiratory center; number three, the kidneys. We will study these systems one by one. Firstly, buffer systems of the body fluids. When there is a change in hydrogen ion concentration, the buffer systems of the body fluids react within seconds. A buffer is any substance that can reversibly bind hydrogen ions. A buffer is any substance that can reversibly bind hydrogen ion concentration. A free hydrogen ion combines with the buffer to form a weak acid that can either remain as an unassociated molecule or dissociate back to the buffer and hydrogen ion. There are three main types of buffer systems of body fluids. Number one, the bicarbonate buffer system is the most important extracellular buffer system of the body. The bicarbonate buffer system consists of a water solution that contains two ingredients. Number one, a weak acid, which is carbonic acid. Carbonic acid ionizes weakly to form hydrogen ion and bicarbonate ions. Number two, the bicarbonate salt. The bicarbonate salt ionizes to form sodium and bicarbonate ions. When a strong acid such as hydrochloric acid is added to the bicarbonate buffer solution, the increased hydrogen ions released from the acid are buffered by the bicarbonate ions as shown in the following equation. Hydrogen ions from the strong acid hydrochloric acid reacts with bicarbonate ions to form very weak acid carbonic acid, which dissociates to form carbon dioxide. This carbon dioxide stimulates respiration and is eliminated from the respiratory passages. When a strong base such as sodium hydroxide is added to the bicarbonate buffer solution according to the following equation, the hydroxyl ions from NaOH combines with carbonic acid, and the weak base NaHCO3 replaces the strong base. The other effects of adding a strong base to the bicarbonate buffer solution are: number one, the concentration of carbonic acid decreases. carbon dioxide levels in the blood decrease. This inhibits respiration and decreases carbon dioxide expiration. Increased blood bicarbonate ion concentration is compensated by increased renal excretion of bicarbonate ions. Phosphate buffer system. The phosphate buffer system has a pK of 6.8. This allows the system to operate near its maximum buffering power. As this is close to 7.4, the main elements of the phosphate buffer system are dihydrogen phosphate and hydrogen phosphate ions. When a strong acid such as hydrochloric acid is added to the phosphate buffer system, the hydrogen is accepted by the base hydrogen phosphate and converted to dihydrogen phosphate according to the following equation. So, the strong acid hydrochloric acid is replaced by a weak acid sodium dihydrogen phosphate. The decrease in pH therefore is minimized. On the other hand, when a strong base such as sodium hydroxide is added to the phosphate buffer system, the hydroxyl ions is buffered by dihydrogen phosphate to form hydrogen phosphate and water according to the following equation. The strong base sodium hydroxide is therefore replaced by a weak base, and there is a slight increase in pH. In contrast to its minor role as an extracellular buffer, the phosphate buffer system plays a major role in buffering the renal tubular fluid because of two reasons. Number one, phosphate becomes concentrated in the tubules, increasing the buffering power of the phosphate system. The tubular fluid has a lower pH than the extracellular fluid nearer to the pK of the phosphate buffer system. Secondly, the phosphate buffer system plays a major role in buffering the intracellular fluid also, because concentration of phosphate is many times in the intracellular fluid than in the extracellular fluid. And the pH of intracellular fluid is also lower than that of the extracellular fluid, closer to the pK of the phosphate buffer system. Proteins as intracellular buffers. Proteins are among the most abundant buffers in the body because of their high concentrations, especially within the cells. Approximately 60 to 70% of the total chemical buffering of the body fluids is inside the cells. Most of this buffering results from the intracellular proteins. Factors contributing to buffering power of proteins include: number one, high concentration of proteins, and number two, pKs of these protein systems are close to intracellular pH. All buffers in a common solution are in equilibrium with the same hydrogen ion concentration. Whenever there is a change in hydrogen ion concentration in the extracellular fluid, the balance of all the buffer systems changes at the same time. This phenomena is called the Isohydric Principal. Respiratory regulation of acid-base balance. The second line of defense against acid-base disturbances is control of extracellular fluid carbon dioxide concentration by the lungs. An increase in ventilation eliminates carbon dioxide from extracellular fluid, which decreases hydrogen ion concentration of the body fluids and vice versa. Changes in either pulmonary ventilation or the rate of carbon dioxide formation by the tissues can change the extracellular fluid partial pressure of carbon dioxide. Increasing alveolar ventilation decreases extracellular fluid hydrogen ion concentration and raises the pH. The higher the alveolar ventilation, the lower will be the partial pressure of carbon dioxide. Increased hydrogen ion concentration stimulates alveolar ventilation, decreasing partial pressure of carbon dioxide in extracellular fluid, leading to a reduction in hydrogen ion concentration back to normal. So, if the pH is suddenly decreased and pH falls from 7.4 to 7.0, the respiratory system can return the pH to a value of about 7.2 to 7.3. Impaired respiration can cause respiratory acidosis. For example, severe emphysema decreases the ability of lungs to eliminate carbon dioxide, causing buildup of carbon dioxide in extracellular fluid, leading to respiratory acidosis. Secondly, impaired respiration can cause respiratory acidosis as the ability to respond to metabolic acidosis is impaired because the compensatory reductions in partial pressure of carbon dioxide that happen by means of increased ventilation does not occur. The third primary system regulating hydrogen ion concentrations in the body fluids is the renal control of acid-base balance. The kidneys control acid-base balance by excreting either acidic or basic urine. Excreting acidic urine reduces the amount of acid in extracellular fluid, and excreting basic urine removes base from the extracellular fluid. The basic mechanism is, if more hydrogen ion is secreted than bicarbonate ion is filtered, there will be a net loss of acid from the extracellular fluid. And if more hydrogen ion is filtered than hydrogen ion is secreted, there will be a net loss of base from the extracellular fluid. Each day, body produces non-volatile acids, mainly from the metabolism of proteins. These acids are called non-volatile because they cannot be excreted by the lungs. They are removed from the body only by renal excretion. The kidneys must also prevent the loss of bicarbonate in the urine. And this is, mind you, quantitatively more important than the excretion of the non-volatile acids. It is important to keep in mind that both the reabsorption of bicarbonate ions and the excretion of hydrogen ions are accomplished through the process of hydrogen ion secretion only by the tubules. Now, this is a very, very important point. It will be discussed in detail in the next lecture. Another key point is that when there is a reduction in the extracellular fluid hydrogen ion concentration, that is alkalosis, kidneys secrete less hydrogen ions and fail to reabsorb all the filtered bicarbonate ions, therefore increasing the excretion of bicarbonate ions. Removal of bicarbonate ions therefore raises the extracellular fluid hydrogen ion concentration back towards normal. The opposite occurs when there is an increase in the extracellular fluid hydrogen ion concentration, which is acidosis. The kidneys secrete additional hydrogen ions. The kidneys do not excrete bicarbonate ions into the urine. They reabsorb all the filtered bicarbonate ions. And the kidneys also produce new bicarbonate ions in addition to the reabsorbed ones, and these events then reduce the extracellular fluid hydrogen ion concentration back towards normal. So these are some of the general points about the renal control of acid-base balance. I stop here and we will continue with the discussion of acid-base regulation in the next lecture.